So, hello, my name is Yasmin, and I was fortunate to be an intern at the Krogan lab through the PUMAS program this summer. And today I'll be talking about G protein coupled receptors and distinguishing their signaling pathway. So, G protein coupled receptors are also shortly referred to as GPCRs, and they are the largest family of membrane receptors responsible for mediating cellular responses to hormones and neurotransmitters. Here's a picture showing how their signaling works once activated by an agonist. They are internalized through endosomes and then can either be digested by lysosomes or recycled back to the plasma membrane. And actually, some GPCRs have been identified to be involved in signaling of involving pain, and one of them being a core kappa opioid receptor, and which has also been established for its analgesia and engaged in chronic pain states, including neuropathic pain. So we want to learn its signaling and understand its trafficking within the cell to help us answer the question for the medical community of how do we possibly target these GPCRs as safer alternatives to opioids. For my project, again, we focus on the kappa opioid receptor, and I use a proximity labeling approach to identify its interactions. And before I get into the workflow, the first step was to generate a cell line expressing our receptor of interest. We started with cloning an apex tag with the receptor in a lentiviral plasmid. This plasmid is then co-transfected with packaging plasmids in order to generate a lentivirus. We then use this lentivirus to infect HEC293 T cells to introduce our apex tag receptor. Next, we characterize the cell line to check the expression of our construct. So diving into the fun part, we have 6 plates, 3 treated with agonist dyne in red and 3 controlled with no agonist. We treat all plates with biotin phenol for a total of 30 minutes, and which helps label the protein surrounding the apex tag receptor, which will help us capture a snapshot of the recruited interactors. Post-labeling, we harvest the cells and collect the lysate. We then purify these biotinylated proteins using structative intact magnetic beams. And to confirm the biotinylation and enrichment of biotinylated proteins, I have a validation Western blot of pre and post enrichment. So pre, we have a lot of proteins, which means we have biotinylated proteins, and then post, we have none, which is good. We then process the enriched samples for mass spectrometry and then compare the list of labeled proteins between control and agonist treated samples. And a huge thank you to Sean Yu for helping gather the mass spectrometry data and help analyze it as well. So this graph serves as a quality control, which has a consistent number of 4,490 proteins all across, meaning we have consistent and reproducible data. And out of those 4,490 proteins, only few were significantly changed, which we can see in the volcano plot. On the right, red, we have upregulated proteins, and blue, we have downregulated proteins. But future directions would be to do a large scale experiments with the receptor at increasing time points, such as 1 minute, 10 minutes, 20, and so on, and including spatial references. After referencing literature for pronounced protein pain interactions, we would then create experiments to see what would happen if we were to inhibit and block this protein. All this would help with understanding their role in pathways in pain signaling and how they can help us develop therapeutics for pain management. A big thank you to my mentor Trupti, as well as others who mentored me along the way. Ruth, Jaewei, Xiaofang, Sean Yu. Individually, thank you. Thank you for helping me persevere in the lab. And thank you to Nevin Krogan for letting me develop my skills in his lab. And thank you to Emma and Sudha for this program. I've had a really good time this summer. And I'll be taking questions now.
Great job, Yasmin.
Thank you.
Questions for Yasmin? Okay.
Biggest challenge? I don't think I had any challenges. I mean, because overall it was all new to me. I didn't know how to do Western blots or apex labeling, or I didn't know about GPCRs. No, it was all fun. I guess sort of designing the experiment and calculations, but I had a really good time. It was awesome. So I don't know if I would call it a challenge. It was just little tests that I passed, got good results.
We like to hear that.
Yeah. Good time.